Okay, good morning, everybody. First of all, I need to start thanking John Francisco. What happened to Brian? Brian, thank you so much for organizing. It really is a pleasure to be here. I feel sort of like this should be the symposium called the phylogeofam or something like that, you know. It's like a family reunion with none of the drama, which is wonderful. So we're here celebrating 30 years of phylogeography. It feels like a birthday party, which is great. And just over the last day, and I'm sure we'll see more of this today, it's been incredible to see the progress that phylogeography has made in the last 30 years. We've embraced genomics, we've embraced all sorts of robust analytical techniques, we are hypothesis testing, we're choosing models. It's just amazing to see the progress. A lot of this progress has come from work on vertebrates, and I'm not being a vertebrate snob, but it is the truth. We saw this in Brett's talk yesterday, and this review from 2008 basically showed the same thing. For the first 20 years of phylogeography, the vast majority of phylogeographic studies, the ones here in asterisks, were performed on vertebrates. Brett's data show that for comparative phylogeography, the pattern is essentially the same for the last 10 years. So what have we learned from all of this? Well, I guess if I were to summarize it all for the vertebrate literature, what we've learned is that refugia are everywhere. And this started not only, this was basically the way phylogeography started from the beginning, was looking for patterns of concordance between responses to environmental variables in terms of phylogeographic distributions. So the earliest paper, Avise et al., 1987, began with this beautiful graph, which is probably engraved in everybody's mind, everybody at least in this room, with patterns in the southeastern US among a number of different vertebrate taxa. That was followed by patterns from the Australian wet tropics, Western Europe, the Atlantic coastal forest, and most recently, different parts of Africa, Eastern and Western Africa. So our search for common pattern undoubtedly has taught us a huge amount about how biodiversity evolves or how it has evolved. But what I'm going to argue today is that what the search for common pattern has done was take our attention away from the search for the not so common pattern. And the reason I think it's important to realize this is because the not so common patterns are often, I'd argue, the more interesting ones because they basically embody cases in which something about the environment or the taxon itself or the group of taxa that are co-evolving, something about them is actually more complex, right? So what I'm going to do today, or what I hope to do today, is to highlight 3 different topics or lessons that I think we've gathered from all our information on vertebrate phylogeography, and talk a little bit about how the exceptions to the common patterns are going to inform the next few years in phylogeographic studies. So those 3 topics are species-specific traits and the independent or idiosyncratic responses in phylogeography. Then I'll show you an example that I think is very interesting about what the concordance between phenotypic and genotypic structure in phylogeography can tell us about mechanisms. And I'll end by giving you an example of the use of functional phenotypes and the phylogeographic distribution of functional phenotypes. So I'm going to be using examples from our group, our lab, and our collaborators, as well as examples from the literature. And you'll notice that all 3 of these topics, in one way or another, address questions at the intersection between the environment, phenotypes, and genotypes. And one of the points I am going to be making is that the environment affects not only the distribution of genotypes, as we know as phylogeographers, but it also has direct impacts on the distribution of phenotypes. And this is another, perhaps, common pattern that, or uncommon pattern that or at least one pattern that we've been ignoring in phylogeography, which is to try to integrate the interactions between genotypes and phenotypes and how we interpret phylogeographic pattern. Okay, so let's start with the first one, species-specific traits and independent or idiosyncratic phylogeographic responses. So we already heard about attempts yesterday by Anna in her work. Brett also alluded to this. But species or unique or idiosyncratic responses to phylogeographic barriers are actually quite common, okay? So although we've been looking for those common patterns, there are always exceptions to every one of those. And we know, just thinking intuitively, that a lot of these differences have to do with differences in the taxa themselves. So these are what we call species-specific traits. So what I would like to do is propose a framework and give you some examples that borrows from the field of biodiversity ecosystem function. So this is a field in ecology called BEF, biodiversity ecosystem function, and our ecologist colleagues are all over this. Basically, in the last 10 years, what ecologists have come to realize, if I go forward here, is that biodiversity is not some sort of epiphenomenon that occurs because of the action of ecosystem and abiotic environments on the distribution of biodiversity. So let me back up a little bit. Traditionally, the way ecologists have viewed biodiversity measures such as species richness or the composition of communities or interactions between species is according to these black arrows, the solid arrows, okay? So traditionally, ecologists have envisioned that the abiotic environment, such as the landscape, rainfall, so forth, dictates the distribution of biodiversity. It also dictates the function of the ecosystems where those different species occur. And those ecosystems, the things that ecologists like to measure, like productivity and biomass and so on, in turn feed back into the distribution of biodiversity. So what the field of BEF actually shows is that biodiversity is inherently part of the process of determining ecosystem function. And so those are these dotted arrows here. So biodiversity, through the traits of the taxa that are part or inherent in those communities, can actually affect ecosystem function through those traits, and ecosystem function can alter the abiotic environment experienced by different members of the communities, and biodiversity can also directly affect abiotic environmental conditions. Okay, so biodiversity ecosystem function, basically what it does is takes the biodiversity aspect or component out of the position of being the outcome, or out of the position of being the response variable, so to speak, and puts biodiversity into a sort of a more active, an active place in terms of this framework. Now, how does this apply to biogeography? Well, we can think of biogeography and phylogeography in a similar way. The difference, of course, is that our function is not ecosystem function. The function we're interested in is evolutionary function. So imagine evolutionary function as a process such as speciation or diversification, turnover of species in various communities. And the triangle is still the same. Okay? Phylogeography has typically focused on the effect of climate or abiotic environmental conditions on the diversification or on the distribution and formation of biodiversity or diversification, and all these dark arrows or solid arrows still apply. So what I'm going to propose is that if we actually adopt this framework and think about traits of individual traits or species-specific traits of taxa, that we can create these alternate arrows just as the ecologists have. So traits, species, with their inherent specific traits, will respond to abiotic environments, of course, but they will also in turn shape the way in which either those species themselves or others in the communities, in the co-distributed communities, are actually diversifying. The ecologists have some names for these different arrows, like ecosystem engineering and niche creation, and many of them may have parallels. So for example, imagine a set of species that are co-evolving, and those species have a particular combined set of traits. And if you were to think about some of those species actually changing or occupying parts of the environment that cause them to then not become available for other species, that's a species interaction, that in turn would alter evolutionary function in terms of altering the exposure of other species in the community to particular environmental conditions. Okay, so it's a framework, something I think worth thinking about, but it basically, as I said before, would allow us to think about the combined effect of species-specific traits on the way that diversification happens, both in the direction and the tempo, as well as the distribution of diversity and the distribution of phenotypes of those traits in the environment. So how far have we actually gotten in terms of thinking about biodiversity in this way in phylogeography? Well, I think we haven't gotten very far. I think we have some baby steps. There are a number of papers out there more recently that have been looking at species-specific traits and the effect of those traits on diversification, not only in that single species, but at the level of communities or co-evolving species. So I think we'll get there. But in general, the studies that have thought about species-specific traits have only done so in a very general way. An example came up yesterday when Brian was talking about, phylogeography of freshwater fishes versus marine fishes, inherently very different. I mean, ultimately, the reason that those are very different is because the species-specific traits of those different groups of organisms in those different environments are actually different. So we have made some progress in thinking about species-specific traits, but generally in these very sort of broad categories. So here's some examples. This is a paper by Lauren Chan, and what she did early on was actually compare the phylogeographic distribution, or the distribution of genetic variation in 2 desert species of frogs, of amphibians, compared to 2 amphibians that live in mesic environments. So these are broad categories, habitat use, but underlying habitat use, of course, is the fact that these animals are adapted, have specific traits to those particular environments. So what you can see here, these are the same 4 lines for the 4 species. This side here, this pointer's not very strong. This one here on your left highlights the desert taxa, and you can see that if you look at isolation by distance, geography by some measure of genetic differentiation, thank you. If you look at geography on the x-axis here by some measure of genetic differentiation, that those lines are relatively flat. And we proposed at the time that these differences between the desert arid environment organisms and the mesic environment organisms had to do with traits that had to do with adaptations to breeding in ephemeral habitats. Okay. More recently, a paper took this one step further. It's a paper by Rodriguez et al. Just this last year in 2015, using 39 different phylogeographic analyses of frogs, mostly in the tropics. What they did was actually look at a number of different traits, both environmental traits, habitat use traits, but in this case also species-specific traits, body size, life history variation, R or K selection, something on the R or K selection continuum, reproductive habitat, and so on. And they use an a posteriori analysis to ask which one of these traits actually affected the depth of phylogeographic diversity. And what they found, and were able to show, very similar to what Lauren showed a few years ago, is that animals living in open environments, non-forested environments, tended to have much shallower phylogeographic signatures than those living in forests. Okay, so again, a habitat use, not necessarily a species-specific trait, but presumably this use of habitat involves also the evolution of particular traits that are contributing to this. So I'm not going to go into much more detail on this because, as I said, I think we're still at the very beginning of thinking about this, but I think that this is really exciting because if we were to adopt this framework, not only can we help explain some of the incongruent results or the independent responses that we have to changes in climate, but also it would allow us to establish sort of a predictive framework. So if maybe we could come up with sets of traits that would be interesting, sets of traits that intersected with particular landscapes would lead us to be able to predict where certain kinds of diversification would happen. So I think that's a very exciting prospect, sort of turning phylogeography on its head in terms of the predictive framework. Okay, the second lesson I want to talk about has to do with concordance between phenotypic and genotypic structure. And this is, my subtitle for this is like the link between phylogeographic lineage structure and phenotypic diversity, it's usually there until it isn't. And the reason that I say that is because I think most of the cases where this phenotypic, genotypic concordance in phylogeographic studies doesn't occur seem to be sort of swept away as not being particularly interesting. Now, the reason that this association between phenotype and genotype I think is important is because it tells us not only about phylogeography itself, but it also tells us more about the mechanisms, and particularly environmental selective mechanisms, that are leading to the origin of biodiversity. And for this topic, I'm going to borrow from the field of genomics of adaptation, evolutionary biology. This has to do particularly with the relative roles of plasticity and adaptive divergence in creating biodiversity. And this has mostly been developed, this framework, in the context of population genetics. There is a really, really nice paper by Erica Crispo from 2008 that I recommend if you're interested in this. And just a couple years ago, Cameron Gallenbohr published a very nice empirical example of this interaction between phenotypic plasticity and adaptive genetic divergence. So let me just sort of give you the big picture. The big picture is that organisms evolving in environments that may differ or not are basically subject to divergent selection, and that's how traits, species-specific traits, evolve. And then they're also subject to other microevolutionary processes such as drift and gene flow and so forth. The big debate centers right here in the middle, which is, the debate is, how do those two microevolutionary processes actually cause the response in terms of change in phenotype to occur? And does that occur through phenotypic plasticity, or does that occur through adaptive genetic divergence? And there are basically, if I summarize it, sort of the big picture, there are basically two possible scenarios. The first is that divergent selection causes organisms that are able to respond plastically to be protected from evolution, because by being able to be plastic, move into certain environments, they basically are not under the same selective pressure as if they did not have plasticity. The alternative is that having a plastic response actually permits organisms to move into environments that they wouldn't be able to, and that then, of course, exposes them to higher selection, therefore furthering adaptive genetic divergence. Okay, so those interested in sort of the genomics and of adaptation are arguing back and forth about whether plasticity actually enhances evolutionary change or whether it deters evolutionary change. And of course, the amount and direction of gene flow and the amount and direction of divergent selection is actually going to be very important in this balance. Okay, we can easily put this into a phylogeographic context because phylogeography is all about gene flow and all about populations being in different environments exposed to divergent selection. So if we think about how this would work in a phylogeographic framework, and just think on your own phylogeographies about the distribution of genes and phenotypes over space for the organisms that you work in. Basically, most of us have a situation that falls into one of these four quadrants. Okay? So along this axis here, I have genetic diversity and whether populations are highly structured or homogeneous. Along this axis here, I have phenotypic diversity and whether that phenotypic diversity is regionally very highly structured or whether it's homogeneous. So in purple here, these two boxes are maybe the least interesting boxes. These are cases where the genetics and the phenotypes match perfectly, okay? So there's a lot of structure, a lot of lineages in your phylogeography at presumably neutral genes. These genes I'm referring to here are the ones we would use to create phylogeographic history for those populations. And the phenotype is highly structured, and they match perfectly, okay? So great. What this tells you is that both phenotypic and genotypic diversity in that particular situation are evolving, perhaps in response to the same mechanisms, barriers to gene flow, drift, selection, so on. Down here, phenotypes are homogeneous and the genetics are homogeneous, so everything's the same. We have no drift and no gene flow barriers, and phenotype and genotype reacting to the same processes. So the interesting ones, of course, are here. These are cases where phylogeographic structure is very high and phenotype is homogeneous, and these are cases where the phylogeographic structure is non-existent or very low, but the phenotypes are highly structured. And the reason these two are very interesting is not only just because of the discordance, but because some of these particular mechanisms need to be happening. So in this case, this is the case where you would find, for example, cryptic species, right? High genetic structure in an otherwise completely similar phenotypic group of populations. And this could happen because the selective environments across this phylogeographic landscape are very similar. There's stabilizing selection or strong uniform selection on the phenotypic traits that you're looking at. Perhaps there's some gene flow barriers that are affecting genetics, but in the face of this stabilizing selection do not affect the phenotypes, and no opportunity for plasticity. Okay? Up here, the mechanisms are the opposite. You can imagine a situation where you had heterogeneous selective environments, very divergent selection on the traits you're looking at, no barriers to gene flow, which means that you're not going to have neutral phylogeographic structure, but a lot of selection that would enhance diversification in phenotypes independent of that homogeneity. And here you have high opportunity for plasticity. Okay, so all the vertebrate studies that actually have looked at some variation in phenotype to genotype fall somewhere here, and I found examples for all of these quadrants, but I think we still don't know, we still haven't done a really systematic survey to figure out where our organisms generally fall. And the reason this is important to know is because this takes us directly to this debate as to whether a plastic response or an adaptive genetic divergence is what's driving most of the diversification, especially in phenotype here. Okay, so I'm not going to talk about cryptic species. We've all seen a number of phylogeographic results, empirical results that show deep diversification with no phenotypic diversification, but I want to talk about a case that's up in this quadrant here. And this is a study that was led by my collaborator, Nick Mason. Nick Mason and Scott Taylor, who are a graduate student and postdoc at Cornell, study red poles. And red poles are a complex of very closely related species. There's 3 of them, apparently, the common, the hoary, and the lesser red pole. And there are 3 species, named as 3 species, and they're phenotypically divergent. I mean, for a bird, these are different, right? I mean, this is streaky, this one's more white, and this one's somewhere in between. So I buy it. Those are 3 different species. A birder would think it's 3 different species. Okay, combined, their distribution is holarctic, and what Nick and Scott did was sample populations from throughout this distribution initially to ask questions about connectivity and space use and distribution of genetic variation. Okay, so they threw at this system pretty much everything they could throw at it. So first they collected rad-seq data, ended up with over 20,000 SNPs across those 3 species. They also used RNA-seq and called SNPs from the RNA-seq data as a second group of markers. And then finally, they also, which I'll tell you about in a second, ended up doing some transcriptomic analysis in terms of variation in gene expression. And this is what they found. It's like absolute homogeneity. Okay? Across the 3 species, the 20,721 SNPs show absolutely no structure at all over that wide distribution. An analysis using SNAP shows that the lineages, this is the outgroup here, these are the 3 red poles, that those lineages are completely overlapping and admixed despite the phenotypic variation, and a principal component analysis of the SNPs called from RNA-seq data show a large overlap as well. So these basically look like one single panmictic population. Environmental niche modeling shows that there is some difference in what you would think in terms of the selective environment, at least for 2 of the species. So environmental niche model for the common red pole shows that it has a slightly larger distribution, at least in this part of the range, and the hoary red pole, the wider one, the whitish one, is actually more limited to northern more northern environments. So there may be some sort of selective difference there. If we examine the degree of phenotypic, genotypic discordance or concordance, this is what we find. So on this side over here, I have, we've listed the phenotypic, genotypic correlation, phenotypes on the y-axis, and some class of marker on the x-axis. And these are anonymous markers, okay? So these are the rad-seq data here at the top. These are the SNPs called from transcriptomes. And what you can see is that there's no association whatsoever. But if you go to the gene expression data, the data that actually look at the differential expression of genes across populations, you see a positive correlation. Okay, so what this tells us is that this is plasticity. Okay, what this tells us is that although you have a genome that is 99% identical across these three species, you can find a correlation, but that correlation, or that variation, that phenotypic variation, is entirely driven by plasticity in an environment that is fairly similar, but in species that are using that environment in slightly different ways, at least according to the environmental niche model. So this is a classic example right here of high plasticity structuring phenotypic diversity, and I think it's one of the few examples showing that extreme of a discordance between phenotypic and genotypic diversity in vertebrates. And I think it's going to be very exciting to try to figure out, using this structure, this discordance matrix, what percentage of the diversity in vertebrates or any other organism, for that matter, what percentage of the diversity that we see in phenotypes is actually due to plasticity versus adaptive divergence. Okay. All right. The last study I want to talk about has to do with using phylogeography to better understand the distribution of functional phenotypes. And for this study, I'm actually going to use an example from my lab that has to do with the evolution of a particular phenotype that we're interested in, and that is the acquisition of resistance or tolerance to a disease. Okay, so in my lab, we have been studying an amphibian disease, and we have a host and a pathogen. Oops, what happened here? Did I tell you the frogs are disappearing? There it is. Okay, so we have a host pathogen system that we have been looking at, and the pathogen is a fungus, a chytrid fungus called Batrachochytrium dendrobetidis, which we call BD. It's a skin fungus. It infects the skin of frogs, and the host that we have been focusing on is the lowland leopard frog, Lithobates javapaensis. This is a pretty bad amphibian disease. It infects a number of different taxa, and some of you may have heard it's responsible for declines and extinctions in a number of different taxa across the world. Okay, so this work was headed up by Anna Savage, who was a student in my lab. She's now a professor at University of Central Florida. And what Anna did for her thesis was actually characterize the geographic distribution of two things: mortality, susceptibility to this fungus, the presence of the fungus in the environment, as well as sort of the genetic underpinnings, the functional variation in terms of the acquisition of tolerance. And what she discovered, first by just doing population surveys, is that there's a clear seasonality to mortality in this system, okay? So the phenotype we're interested in is seasonally modulated, okay? In the winter, in Arizona, where these frogs live, the waters in the streams where these frogs live get cool enough that the fungus explodes in terms of population numbers, and the frogs get sick. In a number of populations, you find dead frogs. So here is the number of frogs in solid lines and the prevalence of the chytrid in the other lines, in the dotted lines, and you can see that there's a clear seasonal alternation. Okay? Every winter, there's huge mortality. Every summer, the temperatures go up in Arizona. The fungus gets brought back down in terms of the fungal population, and the frogs recover, and so on. So that's interesting, seasonal variation, but more interesting is that there's actually spatial variation in the occurrence of this mortality pattern as well. Here's a typical habitat where javapaensis lives in these cool streams. And what Anna found by surveying a number of populations is that we have three different possible syndromes, okay? We have populations here in green where she found no BD in the environment at all, or on the frogs, and of course then there's no BD mortality, and those are these two populations here. We have populations where there is BD present and winter mortality, and those are in red, and that's what you would expect given the fungal disease dynamics. And then the most interesting populations, of course, are these populations in yellow where you have BD in the frogs and you have BD in the environment, but you have no mortality, no winter mortality. And those are these here in yellow. Those populations, of course, are the most interesting in terms of the potential for the evolution of tolerance, which is this phenotype we're interested in. So here's sort of the big picture of what this looks like in the field. This is the prevalence of BD at the populations that we surveyed, and you can see that most of the populations are infected with BD. This is BD mortality, and you see that there are a number of populations that are all white here. Those are the ones where mortality does not exist. And then we also developed, because we knew from previous functional studies, one functional gene to see if we could do a phylogeography of that functional gene as well and superimpose that on the functional phenotype, which is resistance or tolerance. And so we sequenced MHC. MHC is one of the genes involved in the acquired immune pathway. We sequenced a particular part of the MHC which is responsible for binding antigens and starting the production of antibodies in invertebrates. Okay, so how can phylogeography help us here? Well, phylogeography in this case, given this functional phenotype, can tell us a bunch of different things. First of all, if we know what landscape processes in terms of gene flow or drift or selection are occurring in these different populations, phylogeography could potentially inform us of why some of these populations match in terms of their disease dynamics and others don't, okay? So one example, a simple solution, if one were to ask the question, what is keeping these populations from having BD mortality, might be that they are connected by gene flow and those populations have a particular gene that allows for tolerance to BD. So that's one way in which phylogeography could help. The second way in which phylogeography can help has to do with examining the responses to this particular selective environment, which is BD in this case, at different scales. And so one of the advantages of phylogeography is that one can actually look at variation at relatively small scales and compare it to large scales and draw inferences about the relative roles of selection versus other evolutionary forces in terms of the evolution of this particular phenotype. So that's what we did. We created first a phylogeography of all the populations. And what you can see here, right off the bat, if you just look at the structure plot, is that virtually every single population is its own genetic deme, okay? So every population here is a different color, with two exceptions. The first exception is that there is a single deme that includes these two distant populations that are the ones that are BD-free, okay? So these are populations that we presume BD is not there right now, and we presume they have never been exposed to the pathogen. And then the second exception is two populations that are located very close to one another right here, which share this yellow deme, okay? So right off the bat, you can say, well, it's not just gene flow and admixture between populations that is causing disease dynamics to vary among these populations, because every single one of them is independent. So what that means, or most of them are independent. I'll come back to this pair of populations here in just a second. Okay, so what this means is that gene flow isn't contributing to the sharing of resistance alleles or any other kind of allele. So the dynamics in all these populations and diseases should be independent. Can we actually find, by using phylogeography of functional phenotypes or functional genes, can we actually find a signature of that independence in our data? So we basically created a genealogy or a phylogeography of MHC. And the results show that, in fact, we can map functional diversification in terms of tolerance on the same landscape and the same disease dynamics that I just described. So let me walk you through this. This is a genealogy of MHC. It looks messier than when we use nice other gene trees, and that's because there's a huge amount of polymorphism and, of course, a huge amount of overlap in terms of the distribution of alleles in these different populations. The tips here are colored by the populations that we're interested in, and these large pie charts here are the 3 most common MHC alleles in this landscape, allele Q, N, and A. And just to sort of cut straight to the result, what we show is that there is one allele here that is associated with mortality, okay? So it's not that it causes mortality, it's that it's associated with susceptibility, right? So this is a non-tolerant allele, and allele A is present mostly or primarily in populations where we do see mortality, and the relative risk of actually dying or of BD if you have allele A is almost 3-fold baseline, okay? We have another group right here, MHC supertype number 4, which has a relative risk of mortality of 0, so that's a tolerance allele. And then we also have allele Q up here, which falls into MHC supertype number 2, which confers to the populations that have that allele a relative risk of survival of 3 times higher than baseline. Okay, so again, geographic concordance between phenotype and genotype, in this case, the function of this particular, or the expectations of how the distribution of this functional genotype matched the outcome in terms of, our expectations matched the outcome in terms of the performance of those populations in terms of these independent disease dynamics. Okay, just want to end here by telling you a little bit about phylogeography informing functional differentiation at a different scale. And that's because I think this story here is kind of great. These 2 populations right here are in a locality called Muleshoe Ranch. Muleshoe Ranch is a little bit unique for Lithobates yavapayensis because one of the streams at Muleshoe Ranch is actually fed by a hot spring, okay? And so one of the localities actually has, even though this is over a very short geographic scale, one of the localities actually has a very, very different selective environment. Okay, so the hot spring sits right here in this little map, and when Anna realized, the reason that we went back to sample more is that Anna realized that this population was unique and that it was showing the mortality you would have expected from a population that had a lot of that big susceptibility allele, but the resistance allele was actually present in this population. So the way she noticed this is that there was a discordance between the expectations in terms of the genotypic distribution and the phenotypic outcome of disease dynamics. So when she went there to sample more, what she found is that there's frogs living here in this hot spring population where the water is only 2 to 3 degrees warmer, but that change in the selective environment completely changes the exposure of the frogs to BD. Okay? And then down here in Bass Canyon, just the next stream down, you have a population of frogs that is dying all the time, has mortality in the winter, even though they have the presence of this resistance allele, of this tolerance allele. And so what Anna discovered is that basically what's going on here is that this population is sheltered from selection for tolerance. Okay, so this population, because of the warm water, never is selected for tolerance. They don't have to be. Okay, the selective coefficient here for evolving tolerance is 0 because there is no fungus in that warm water. Okay, and so what happens is that these animals die. There's unidirectional migration from this population to this one, and what this means is that there's a constant influx of susceptibility alleles, even though there's strong selection for the evolution of resistance in this other population. So again, at a very different scale, very small scale in this case, but it's a clear example of how phylogeography and functional phylogeography can actually inform the mechanisms that are underlying the outcome in terms of phenotype. Anna also did some calculations to figure out how much selection would it actually take in this receiving population to evolve resistance, and it turns out that the number is relatively high. So you would have to have huge selection for tolerance in this population to undo this constant flux of susceptibility alleles that is coming from the non-selected population. Okay, so what I've tried to convince you of here, hopefully successfully, is that one of the areas where I think phylogeography needs to go is to sort of try to reintegrate phenotypes into our understanding of the distribution of variation. There are a number of advantages to doing that. One is that this helps us understand evolutionary function, as I described, and that evolutionary function is what underlies the origin and the loss of biodiversity. It helps us discover environmental and genetic controls and the relative contribution of environmental and genetic controls to the distribution of functional phenotypes. This will be important for conservation. Phenotypes are what are going to respond in terms of selection to global change. It's what we're going to see in terms of responses, in terms of resilience or adaptive potential. And so I think that's an important aspect as well. And it's the reason that we do phylogeography, right? I mean, I think most of us, if you do phylogeography, you're interested in biodiversity. When you open a tray of specimens or you go to the field and you see variation, you don't see mitochondrial DNA floating around in space. What you see is the diversity itself. So not only in terms of understanding historical selection, but also understanding the origin of this diversity itself. Okay. I'm going to stop here. Thank you very much. I want to say thank you to my funding sources.